Hello there, and welcome back to Bedtime Science. I am really glad you are here tonight. At this moment, the body may appear to be doing very little. The room is quiet. The hands may be still. Breathing continues without instruction. The heart repeats a rhythm it has already performed billions of times. Yet inside almost every tissue, maintenance is underway. Enzymes move along DNA and correct chemical damage. Proteins that have folded incorrectly are refolded, dismantled, or carried towards cellular recycling systems. Immune cells inspect tissue. Stem cells divide when replacement is needed. Mitochondria merge, separate, and remove damaged parts. Bone is dissolved in microscopic regions and rebuilt. Blood cells that have reached the end of their useful lives are cleared while new ones enter circulation. The body is not a structure built once and then slowly worn down. It is an activity. A human body remains recognizable across decades even though many of its molecules and cells are replaced. Identity persists through regulated change. Aging begins inside that achievement. It is not simply the presence of damage. Damage begins before birth and continues every day. It is not simply the loss of cells. Cell loss can be useful, and uncontrolled survival can become cancer. It is not simply the shortening of telomeres, the slowing of mitochondria, inflammation, or a clock hidden in DNA. Aging emerges when many systems of maintenance, repair, replacement, and communication gradually lose the coordination that once kept the whole organism resilient. The loss is uneven. The lens of the eye contains proteins that can remain for a lifetime. The intestinal lining renews rapidly. Neurons in much of the brain may survive for many decades. Blood is replenished continuously. Bone appears solid but is remodeled throughout life. A person does not age at one rate in every tissue. One artery can stiffen while muscle remains strong. Immune responses can weaken while memory remains clear. The skin can show decades of sunlight that internal organs never received. A joint injured in youth can become older in function than the rest of the body. Chronological age is shared by every cell. Biological history is not. Tonight, we will follow that history slowly. We will begin with a mathematical curve drawn in the early 19th century, when a man studying mortality noticed that the risk of death did not rise in a straight line. We will move into the evolutionary puzzle of why bodies are repaired so well and yet not forever. We will enter cells that can divide only a limited number of times, chromosome ends that shorten, proteins that lose shape, mitochondria that carry ancient bacterial ancestry, and immune signals that protect in youth but can become persistent in later life. We will meet animals that make ordinary human aging look surprisingly fast. A clam that began life centuries before it was collected. A shark moving through Arctic darkness for perhaps hundreds of years. A small underground rodent whose mortality pattern barely follows the rule expected for mammals of its size. We will return to human beings through centenarians, disputed longevity regions, epigenetic clocks, calorie restriction, exercise, rapamycin, metformin, senolytics, NAD precursors, and experiments that attempt to make old cells behave as though they were younger. Some findings are solid. Some are promising in mice. Some alter biomarkers without proving that anyone will live longer. Some have grown into industries far ahead of the evidence. The aim is not to promise escape from aging. It is to understand why aging is not one process, why no single switch controls it, and why the most meaningful scientific goal may be less about adding years at any cost than preserving function within the years that exist. The story begins with death records. In 1825, Benjamin Gompertz published an essay on human mortality. Gompertz was a self-taught mathematician born into a Jewish family in London at a time when religious restrictions limited his access to universities and learned societies. He studied mathematics independently and later worked in insurance. Insurance companies needed to estimate risk across age. A policy written for a young adult carried a different probability from one written for an older person. Mortality tables existed, but Gompertz looked for a mathematical pattern beneath them. He noticed that adult mortality did not merely add the same amount of risk each year. It rose approximately exponentially across much of adulthood. In a simplified form, the chance of dying during a given interval increased by a roughly constant proportion over time. Later summaries often described human mortality as doubling about every 7 or 8 years through much of adult life. Though the exact rate varies across population, era, sex, health, and method, the curve became known as the Gompertz law. It does not explain aging. It describes one of its consequences. A straight line would imply that each year adds the same hazard. The Gompertz curve implies acceleration. The older organism becomes vulnerable not only because another year has passed, but because the systems that resisted previous years have themselves changed. Damage can make future damage harder to repair. Loss of reserve makes the next stress more consequential. A young body can respond to infection, fracture, blood loss, or sleep deprivation and return near its previous state. An older body may recover more slowly or settle at a lower level of function. This capacity to return is sometimes called resilience. Aging can be understood partly as the narrowing of resilience. The resting heart may still beat normally. The blood test may appear acceptable. Then a stress reveals the reduced margin. An illness lasts longer. A fall produces a fracture. A medication causes confusion. A period of inactivity leads to muscle loss that is difficult to regain. The organism can maintain ordinary life while becoming less able to absorb disturbance. Gompertz saw the population curve. Biology would spend the next 2 centuries asking what creates it. One possibility seems obvious: wear and tear. A machine accumulates friction, corrosion, cracks, and loosened parts. A body also accumulates molecular damage. Oxygen chemistry alters proteins and DNA. Mechanical load changes cartilage. Ultraviolet light injures skin. Toxins and infections leave traces. The analogy is useful until it becomes too literal. Machines do not grow from one cell. They do not replace parts from within, monitor their own errors, reproduce, heal wounds, or alter maintenance according to food and danger. A body is built from components that are constantly being dismantled and rebuilt. Aging is therefore not ordinary where acting on a passive object. It is where acting on a self-repairing system whose repair priorities were shaped by evolution. This leads to the central evolutionary puzzle. Why did natural selection not produce bodies that repair indefinitely? An immortal organism might seem to have an advantage. It could survive, reproduce repeatedly, and preserve everything learned. But natural selection does not design for indefinite future survival. It favors traits through their effects on reproductive success within real environments. In the wild, animals die from predation, infection, starvation, competition, weather, injury, and chance. A mutation that improves survival or reproduction early can spread even if it causes harm late, especially when few individuals reach the age at which the harm appears. The force of natural selection weakens with age after reproduction begins. This insight became the foundation of evolutionary theories of aging. In the mid-20th century, Peter Medawar described what became known as mutation accumulation. Imagine a harmful genetic variant whose effect appears before reproduction. Individuals carrying it are less likely to leave offspring. Selection removes it strongly. Now imagine a variant whose harm appears only at an age rarely reached in the ancestral environment. Most carriers have already reproduced before the effect matters. Selection against it is weak. Across generations, many late-acting harmful variants can remain or accumulate. Aging in this view is not one adaptation. It is partly the shadow created where selection becomes less effective. George C. Williams extended the idea through antagonistic pleiotropy. Pleiotropy means that one gene can affect several traits. A variant may strengthen bone growth, accelerate development, increase fertility, or improve immune defense early in life while increasing cancer, inflammation, or tissue damage later. If the early benefit improves reproduction enough, selection can favor the variant despite the later cost. The same biological pathway can therefore protect youth and burden old age. Growth signals provide a clear example. A young organism must build tissue rapidly. Cells must divide. Proteins must be synthesized. Nutrients must be directed toward growth. The pathways that support this expansion are essential. Persistent activation later can contribute to cancer, metabolic disease, and reduced cellular recycling. Inflammation provides another example. A rapid inflammatory response can control infection and repair injury. The same machinery, activated chronically, can damage vessels, joints, brain, and metabolism. Blood clotting prevents death from injury. Excess clotting later can cause stroke or heart attack. Aging biology is filled with functions whose value depends on timing. Thomas Kirkwood later proposed the disposable soma theory. The word soma refers to the body apart from the germ line that produces offspring. Energy is limited. An organism must allocate resources among growth, movement, defense, reproduction, and repair. Perfect maintenance would be costly. If an animal is likely to be eaten within a year, investing enough energy to preserve its body for 50 years provides little evolutionary return. Selection favors maintenance that is adequate for expected survival, not infinite. This does not mean the body is carelessly built or literally discarded after reproduction. Humans often survive and contribute to family and community long after direct reproduction. Older individuals can increase the survival of descendants through knowledge, food, care, and social support. The theory describes trade-offs, not a death program. Aging is not a switch that evolution turns on at a chosen birthday. It is what can emerge when maintenance is good enough, but not perfect enough forever. Species reveal how strongly ecology changes the trade-off. Mice face predation and reproduce quickly. In the wild, many die young. Their bodies are not selected to maintain function across a human lifespan. Bats are similar in size to mice, yet can live far longer. Flight reduces many forms of predation and allows escape from danger. A longer-lived body can produce more offspring across many seasons, making investment in maintenance more valuable. Birds also tend to outlive terrestrial mammals of similar size. Tortoises protected by shells, whales protected by size, and animals living in stable, deep environments can evolve slower life histories. The rate of aging is not dictated by chemistry alone. It is shaped by the probability of reaching tomorrow. Humans inherited a particular compromise. Our species develops slowly, invests heavily in each child, learns for years, and depends on cooperation. Human lifespan is long compared with most mammals of similar size, especially among primates. Yet, even human maintenance eventually loses precision. The loss does not begin in one organ. It begins across a network of small changes whose interactions become visible only with time. In 2013, researchers organized many of these changes into 9 hallmarks of aging. A 2023 update expanded the framework to 12. Genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, disabled macroautophagy, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, altered intercellular communication, chronic inflammation, and dysbiosis. The hallmarks are not 12 independent clocks. They are 12 views of one interacting system. DNA damage can produce senescence. Senescent cells can produce inflammation. Inflammation can impair stem cells. Mitochondrial dysfunction can increase stress signals. Failure of autophagy can leave damaged mitochondria in place. Changes in the microbiome can alter immune communication. The categories help scientists design experiments. The body does not experience them as categories. It experiences a network becoming harder to stabilize. The first part of that network is already active tonight. Inside a cell, DNA is being damaged and repaired. The damage is ordinary. The repair is ordinary. Aging begins not when either process appears, but when their balance slowly changes. DNA is chemically stable enough to preserve information and chemically vulnerable enough to require constant repair. Water can alter bases. Reactive molecules can break strands. Ultraviolet light links neighboring bases incorrectly. Replication introduces occasional errors. Smoke, radiation, inflammation, and ordinary metabolism add more. A human cell experiences many DNA lesions each day. Most do not become permanent mutations. Repair enzymes recognize distorted structures, remove damaged sections, copy intact templates, and reconnect strands. Several repair pathways specialize in different problems. Base excision repair removes small chemical changes. Nucleotide excision repair removes bulky distortions such as those caused by ultraviolet light. Mismatch repair corrects errors left after replication. Double-strand breaks can be rejoined directly or repaired using a matching template. The system is impressive and imperfect. Some lesions escape. Some repairs introduce changes. A cell lineage gradually acquires mutations. This creates a mosaic body. Two neighboring cells that began from the same embryo can carry different mutations decades later. Skin exposed to sunlight contains clones with mutations associated with cancer even when the skin appears normal. Blood stem cells can acquire mutations that allow one lineage to expand more than others, a condition called clonal hematopoiesis. Most acquired mutations do not produce obvious harm. Some alter cell behavior. A rare combination can support cancer. Others may reduce function subtly or change how a tissue responds to stress. Genomic instability is therefore not one pile of random errors counted at death. It is an evolving population of cells competing and cooperating inside the body. The genome is protected not only through repair, but through structure. DNA is wrapped around proteins called histones and folded into chromatin. Some regions remain accessible to genes that need to be active. Others are compacted and quiet. With age, this organization can drift. Genes may be expressed at the wrong level or in the wrong context. Repetitive elements that were kept silent can become more active. Boundaries between chromatin regions weaken. Cells retain their identity less precisely. This is part of epigenetic aging. Epigenetics refers to changes in gene regulation and chromatin state that do not alter the DNA sequence itself. Methyl groups attached to DNA can affect gene activity. Histone modifications change chromatin accessibility. Regulatory proteins and non-coding RNAs coordinate which genes are used. A liver cell and neuron contain nearly the same genome, but read different parts. Aging can make that reading less stable. The cell does not forget completely that it is a liver cell or neuron. It becomes noisier. Some genes associated with stress remain activated. Repair programs become less responsive. Inflammatory signals persist. Developmental pathways appear where they no longer belong. The idea that aging includes loss of epigenetic information has inspired attempts to reset old cells. But before reaching those experiments, the chromosome ends offer a more concrete story. Every linear chromosome faces a copying problem. DNA polymerase works in one direction and requires a starting point. At the very end of a chromosome, the replication machinery cannot copy all the way to the final nucleotide on one strand. Without protection, chromosomes would shorten into important genes with every division. Telomeres solve part of the problem. Human telomeres consist of repeated DNA sequences and associated proteins at chromosome ends. They act as expendable buffers and prevent the ends from being mistaken for broken DNA. They are often compared with plastic tips on shoelaces. The comparison captures protection, but telomeres are more active than plastic caps. They form loops, recruit proteins, regulate repair access, and participate in signaling. In many somatic cells, telomeres shorten as cells divide. When they become critically short or damaged, the cell can activate checkpoints and stop dividing. This protects the genome. A cell with unstable chromosome ends should not continue copying itself. The cost is reduced regenerative capacity. The connection between limited division and aging entered science through cell culture. In the early 1960s, Leonard Hayflick and Paul Moorhead grew normal human fibroblasts in laboratory dishes. At the time, many researchers believed that cultured cells could divide indefinitely if provided with the right conditions. Earlier work had promoted the idea of cellular immortality. Hayflick found something different. Normal human cells divided a finite number of times, often around several dozen population doublings, then entered a state of permanent growth arrest. The cultures were not simply contaminated or starved. Young frozen cells resumed division after thawing and used the remaining part of their division capacity. Cells taken from older donors often showed less capacity than cells from embryos, though the relationship in living tissues is more complex than a simple counter. The phenomenon became known as the Hayflick limit. Cancer cells behave differently. Many could divide indefinitely. The contrast suggested that mortality at the cellular level could protect mortality at the organism level. A normal cell stops rather than risk copying damaged chromosomes forever. A cancer cell escapes the stop. Telomere shortening later provided part of the mechanism. In the 1970s, researchers including Alexei Olovnikov proposed that chromosome ends would shorten during replication and might limit cellular division. The structures themselves had been studied earlier in single-celled organisms. Elizabeth Blackburn examined the chromosome ends of Tetrahymena, a ciliate with many small linear chromosomes. She found simple repeated sequences. Jack Shostak showed that these sequences could protect linear DNA introduced into yeast. The repeated ends worked across species. A major question remained. Some cells divide repeatedly across generations. Germ cells produce descendants without losing chromosome ends completely. Certain single-celled organisms maintain telomeres despite constant division. An enzyme had to rebuild them. Carol Greider began searching for it as a graduate student in Blackburn's laboratory. She used extracts from Tetrahymena and synthetic DNA substrates, looking for evidence that something in the cells could add telomere repeats. For months, the results were uncertain. On Christmas Day in 1984, Greider saw a ladder-like pattern on a gel. The extract had extended the DNA in repeated units. She and Blackburn named the enzyme telomerase. Telomerase carries an RNA template and uses it to add telomeric DNA. It is active in germ cells, some stem cells, activated immune cells, and many cancers. Most ordinary somatic cells keep activity low. This creates another evolutionary compromise. More telomerase could preserve regenerative capacity. It could also help damaged cells divide without limit. Most human cancers activate telomerase or another mechanism for maintaining telomeres. Telomere shortening can contribute to tissue aging and disease. Inherited defects in telomere maintenance cause syndromes involving bone marrow failure, lung fibrosis, liver disease, immune dysfunction, and premature tissue decline. Yet simply lengthening telomeres is not a universal anti-aging solution. Very long telomeres can increase the window in which precancerous cells acquire additional mutations. Genetic studies show complex relationships between longer telomeres, lower risk for some degenerative diseases, and higher risk for several cancers. The same structure participates in both regeneration and tumor suppression. Average telomere length also makes a poor personal countdown clock. It differs among tissues. People begin life with different lengths. Measurement methods vary. Immune cell composition changes blood results. Stress, infection, smoking, and inherited factors influence averages. Short telomeres can correlate with disease and mortality across groups without predicting one individual's remaining lifespan. A biological association is not a calendar. Cells can become senescent for reasons beyond telomeres. DNA breaks, oncogene activation, oxidative stress, mitochondrial dysfunction, and tissue injury can all trigger the state. Senescence is often described as permanent arrest, but the word permanent hides diversity. Senescent cells differ by tissue and trigger. Some remain metabolically active. Some resist cell death. Some are cleared quickly. Others persist. They can be useful. During embryonic development, senescent-like cells help shape tissues. After injury, temporary senescence can limit proliferation, recruit immune cells, and assist wound repair. It can stop a damaged cell from becoming a tumor. The problem arises when cells accumulate and communication changes. Many senescent cells release a mixture of cytokines, growth factors, enzymes, and other signals called the senescence-associated secretory phenotype. These signals can attract immune cells for clearance. They can also spread inflammation, alter neighboring cells, weaken tissue structure, and promote further senescence. A local emergency signal becomes chronic background noise. With age, immune clearance may become less efficient. New senescent cells continue to appear. The balance shifts from temporary repair toward persistence. This made senescent cells attractive targets. In mice, genetic methods that remove selected senescent cells can delay or reduce several age-related problems. Drugs called senolytics attempt to kill senescent cells selectively. Xenomorphics attempt to reduce their harmful secretions without removing them. The animal results are important. Human evidence remains early. Small trials have tested combinations such as dasatinib and quercetin in specific diseases. Some show biological activity or changes in markers. They do not yet prove that senolytics slow normal human aging or extend life. Removing every senescent cell would not necessarily be desirable. Some support tissue structure or healing. A treatment must distinguish harmful persistent cells from useful temporary states. The cell that refuses to divide is not simply old debris. It may be preventing cancer, holding a wound, signaling danger, or causing damage. Aging repeatedly turns on this ambiguity. Protection at one time can become burden at another. The genome is repaired to preserve information. Division is stopped to preserve stability. Telomeres shorten partly because unlimited division is dangerous. A cell can age to protect the organism, while enough aging cells can harm the organism. No single direction is entirely beneficial. The body survives through balance, and balance becomes harder with time. A cell contains more than DNA. It contains thousands of kinds of proteins, each folded into a shape that allows a task. Some form channels through membranes. Some carry oxygen. Some act as enzymes. Some pull chromosomes apart during division. Some create muscle contraction. Some become antibodies. Some organize the internal skeleton of the cell. A protein begins as a chain of amino acids. The chain bends, twists, and settles into a three-dimensional structure. The final shape depends on its sequence, the surrounding chemistry, helper proteins, and modifications made after synthesis. Folding can fail. Heat, oxidation, mutation, acidity, and mechanical stress can destabilize proteins. A damaged protein may lose function, expose sticky regions, and bind to other proteins. Cells therefore maintain proteostasis, the control of protein production, folding, transport, and removal. Molecular chaperones help proteins fold or recover after stress. The ubiquitin proteasome system labels many damaged or short-lived proteins and feeds them into a molecular barrel for destruction. Lysosomes break down larger structures. Autophagy encloses damaged components inside membrane compartments and delivers them to lysosomes for recycling. The word autophagy means self-eating. It can sound destructive. In practice, it is maintenance. A cell dismantles worn components and reuses amino acids, lipids, and other materials. During starvation, autophagy supplies fuel. During ordinary life, it removes damaged mitochondria, protein aggregates, invading microbes, and cellular debris. Selective forms target particular structures. Mitophagy removes mitochondria. Agrophagy helps remove protein aggregates. Xenophagy targets some pathogens. With age, several parts of proteostasis become less efficient. Chaperone responses can weaken. Proteasome activity changes. Lysosomes become altered. Autophagic flux can decline. Damaged proteins remain longer. In neurons, which may survive for a lifetime and cannot simply be replaced easily, the problem is especially important. Alzheimer's disease includes accumulations of amyloid beta outside cells and altered tau protein within neurons. Parkinson's disease involves alpha-synuclein aggregates in many cases. Huntington's disease arises from an expanded protein sequence that promotes misfolding and toxicity. These diseases are not normal aging, and their causes are more complex than dirty cells failing to take out waste. Age raises risk partly because protein quality control, inflammation, vascular health, and cellular reserve change. Aging makes the nervous system less able to tolerate a process that a younger tissue might contain. Proteostasis also connects with nutrition. When nutrients are abundant, cells receive signals to grow, synthesize proteins, and store energy. When nutrients are scarce, cells slow growth, mobilize reserves, and increase maintenance. The central pathways are ancient. Insulin and insulin-like growth factors signaling report energy and growth conditions. AMP-activated protein kinase, or AMPK, responds to low cellular energy. Sirtuins depend on NAD and regulate metabolism, stress responses, DNA repair, and other processes. The mechanistic target of rapamycin, mTOR, integrates amino acids, growth factors, energy, and stress. mTOR is not an aging switch. It is a coordinator. During development, it supports growth. In adult tissues, it helps muscle respond to exercise, immune cells activate, wounds heal, and proteins are made. Persistent or excessive signaling can suppress autophagy and contribute to metabolic dysfunction, cancer, and aspects of aging. Too little signaling can impair immunity, healing, and muscle maintenance. The pathway became famous through a molecule found in soil from Rapa Nui. In the 1960s, a Canadian scientific expedition collected soil and biological samples from the Pacific Island, also known as Easter Island. Researchers later isolated a compound produced by a soil bacterium. The compound had antifungal activity and was named rapamycin after Rapa Nui. Its story moved far beyond fungi. Rapamycin suppresses immune responses and became useful in organ transplantation. It inhibits abnormal cell growth and is used in certain cancers and medical devices. In yeast genetics, resistance to rapamycin led researchers to proteins called targets of rapamycin, or TOR. The mammalian pathway became mTOR. A molecule collected during one expedition had opened a route into one of the oldest systems controlling growth and maintenance. The decisive mammalian test was designed to resist one laboratory's luck. The National Institute on Aging created the Interventions Testing Program to test proposed longevity compounds in genetically heterogeneous mice at 3 independent sites. The same intervention would be used under closely matched conditions in Maine, Michigan, and Texas. A result appearing in all three places would be harder to explain through one mouse strain, one room, or one local laboratory practice. Rapamycin created an unexpected problem before the experiment properly began. The compound was unstable in ordinary mouse food. Researchers developed a microencapsulated formulation that could survive preparation and release the drug after the animals ate it. The delay meant that the mice were already about 600 days old when treatment started, roughly 20 months of age. A study intended to test lifespan extension had accidentally become a test of late intervention. In 2009, the interventions testing program reported that rapamycin extended median and maximal lifespan in both male and female genetically heterogeneous mice. The effect appeared across all three sites. The result was important not because an old mouse became young. It showed that changing mTOR signaling after much of the lifespan had already passed could still alter survival. The mice were about 20 months old when dosing started, an age often compared loosely with late middle age in humans. The result was important because the treatment had not begun during development. It altered survival after aging was already underway. Later mouse studies tested different doses, timing, and combinations. Rapamycin repeatedly extended lifespan in several settings, though effects varied by sex, strain, schedule, and side effects. This is strong evidence in mice. It is not proof in people. Human rapamycin research has examined immune function, skin, periodontal disease, ovarian aging, and other outcomes. Small trials of low or intermittent doses suggest that some regimens can be tolerated and may alter selected biomarkers. There is no completed trial demonstrating that rapamycin extends human lifespan. A 2026 study in older adults also raised a practical caution. Low-dose rapamycin taken during an exercise program appeared to blunt some improvements in physical performance and produced more side effects in that small trial. The same pathway that may promote maintenance when restrained is required for adaptation to exercise. Timing matters. Dose matters. Tissue matters. A treatment that helps one process can interfere with another. Calorie restriction reaches many of the same pathways without one drug. In the 1930s, Clive McKay and colleagues fed rats diets containing fewer calories while maintaining essential nutrients. The restricted animals grew more slowly and lived longer than animals allowed to eat freely. The finding became one of the most repeated results in aging research. Calorie restriction extends lifespan in yeast, worms, flies, rodents, and some other model organisms under many conditions. It alters insulin and IGF signaling, AMPK, mTOR, sirtuins, autophagy, inflammation, body temperature, and reproduction. The intervention does not reveal one mechanism. It changes the organism's entire allocation between growth and maintenance. The effect also varies. Different mouse strains respond differently. Some gain lifespan. Some show little benefit. A few can do worse under particular diets and conditions. Laboratory animals live in controlled environments without the same infections, predators, social conditions, and food uncertainty as wild animals. A diet that extends life in a cage may reduce survival in a harsh environment. Two long studies of calorie restriction in rhesus monkeys produced results that initially seemed conflicting. One found improved health and survival. The other found health benefits without a clear overall survival extension. The monkeys differed in diet composition, feeding practices, genetics, and the comparison group. In one study, control animals ate freely and became heavier. In the other, controls were not as overfed. Calorie restriction can look more powerful when compared with excess. This distinction matters in human life. Reducing overnutrition is not the same experiment as pushing a healthy, lean person into chronic energy deficit. The calorie trial tested sustained calorie restriction in healthy adults without obesity. Participants were assigned to reduce calorie intake for 2 years, with a target greater than most achieved in practice. The intervention improved several cardiometabolic risk factors. A 2023 analysis found a small slowing in the pace of aging measured by 1D in a methylation algorithm, Dunedin pace. Other epigenetic age measures did not show significant rejuvenation. The effect was measurable and modest. The trial did not show that participants became biologically younger in every sense or that they would live longer. Long-term calorie restriction can also reduce bone density, lower sex hormones, cause cold intolerance, reduce muscle if protein and resistance training are inadequate, and become psychologically difficult. A mechanism associated with longevity is not automatically a safe lifestyle instruction. Intermittent fasting and time-restricted eating attempt to separate periods of feeding from periods of low nutrient signaling. In animals, some benefits occur even when total calories are similar, suggesting that meal timing and circadian biology matter. In humans, time-restricted eating can improve weight and metabolic markers for some people, especially when it reduces late eating or overall intake. Trials vary in duration, eating window, population, and calorie matching. No fasting pattern has been shown to extend maximum human lifespan. The strongest human evidence remains about specific metabolic outcomes, adherence, and who may be harmed. Pregnancy, eating disorders, frailty, diabetes medications, and other conditions can make fasting unsafe. The biology is real. The marketing often outruns it. Sirtuins followed a similar path. In yeast, the protein SIR2 became linked with gene silencing and lifespan under some experimental conditions. Mammals have 7 sirtuins with roles in nucleus, cytoplasm, and mitochondria. They depend on nicotinamide adenine dinucleotide, NAD, a molecule central to energy metabolism and cellular signaling. NAD levels change with age in several tissues and species. This led to interest in precursors such as nicotinamide riboside and nicotinamide mononucleotide. In mice, supplementation can improve selected metabolic, muscular, vascular, and neurological measures under certain conditions. Human trials reliably show that oral precursors can raise NAD-related metabolites in blood. The next step is less clear. Most trials are small and short. Functional benefits are inconsistent. Raising a molecule does not prove that aging has slowed. The tissue that needs NAD may not receive the relevant increase. Metabolism may adapt. Effects may depend on deficiency or disease. By 2026, reviews of human evidence still described clinical anti-aging benefits as inconclusive. The same is true of popular sirtuin activators such as resveratrol. Interesting pathways do not guarantee effective supplements. A cell's maintenance state depends on energy, but aging is not cured by permanent starvation signals. Growth and repair alternate. Muscle needs protein synthesis after use. Immune cells need expansion during infection. Skin needs proliferation after injury. Autophagy needs periods of activity, not continuous dismantling. Youth is not defined by maximal mTOR or minimal mTOR. It is defined partly by flexibility, the ability to switch appropriately. After food, build. During scarcity, recycle. After exercise, adapt. After injury, repair. When the signal ends, return toward baseline. Aging can reduce this flexibility. Some pathways remain activated too long. Others fail to respond strongly enough. The cell becomes less able to match state with need. The problem is not merely too much growth or too little recycling. It is a loss of timing across the system. Mitochondria sit near the center of this changing flexibility. They are often called the powerhouses of the cell because they produce much of the ATP used for cellular work. The phrase is accurate and incomplete. Mitochondria also regulate calcium, synthesize molecules, respond to nutrients, generate heat, signal stress, and help decide whether a damaged cell should die. They carry their own small genomes because their ancestors were once free living bacteria. More than 1 billion years ago, one cell entered into a lasting partnership with another. The internal bacteria became organelles. Most of their genes moved into the host nucleus, but a small mitochondrial chromosome remained. Every breath now depends on that ancient union. Electrons move through protein complexes in the inner mitochondrial membrane. Their energy pumps protons across the membrane. The gradient drives an enzyme that produces ATP. The system is efficient and not perfectly sealed. Electrons can escape and react with oxygen, producing reactive oxygen species. For decades, one dominant theory proposed that these molecules damage mitochondria, which then produce more reactive species, creating a self-amplifying cycle of aging. The free radical theory captured a genuine process but became too simple. Reactive oxygen species are not only damage. At controlled levels, they act as signals. Exercise temporarily increases oxidative stress and triggers protective adaptation. Cells use redox chemistry in immunity and metabolism. Antioxidants supplied in large doses do not reliably extend life and can interfere with beneficial responses. The issue is not the complete elimination of oxidation. It is control. Mitochondria are dynamic. They fuse, allowing contents to mix. They divide, helping distribute themselves in separate damaged portions. Mitophagy removes mitochondria that cannot maintain membrane potential or function. New mitochondria are produced according to demand. With age, these quality control systems can change. Mitochondrial DNA mutations accumulate in some cells. Energy production becomes less adaptable. Damaged organelles release DNA or other molecules into the cell. Because mitochondrial DNA resembles bacterial DNA, its appearance in the wrong place can activate immune sensors. A fragment from an ancient symbiont can be interpreted as infection. This connects mitochondrial dysfunction with inflammation. Inflammation is not one disease. It is a coordinated response to damage, microbes, toxins, and stress. Blood vessels change. Immune cells move. Chemical signals amplify defense. Tissue becomes warmer, swollen, painful, or altered. Acute inflammation is essential. Without it, a wound would not be protected and many infections would spread. Chronic low-grade inflammation is different. With age, inflammatory markers often rise even without one obvious infection. The pattern has been called inflammaging. Several sources contribute. Senescent cells release inflammatory signals. Visceral fat can produce cytokines. Damaged mitochondria and cellular debris activate innate immune pathways. The intestinal barrier and microbiome change. Repeated infections leave immune history. Air pollution, smoking, poor sleep, inactivity, periodontal disease, and chronic illness add further signals. The immune system itself ages. The thymus, where T cells mature, begins shrinking after childhood and gradually replaces much of its active tissue with fat. Fewer new naive T cells enter circulation. The existing population becomes shaped by decades of encounters. Some clones expand strongly in response to persistent viruses. The repertoire narrows. Responses to new pathogens and vaccines can become weaker, while inflammatory signaling remains elevated. This combination is called immunosenescence, though the immune system does not simply shut down. Some functions decline. Some become dysregulated. Autoimmune reactions can rise. Cancer surveillance changes. Wound repair slows. Old immune memory remains useful, while new adaptation becomes harder. Blood production changes at its source. Hematopoietic stem cells in bone marrow generate red cells, platelets, and immune lineages. Across life, individual stem cells acquire mutations. Some mutations give one clone a growth advantage. The clone contributes an increasing fraction of blood cells. This age-associated clonal hematopoiesis can occur without leukemia or symptoms. It also increases the risk of blood cancers and cardiovascular disease in some people, partly because altered immune cells can promote inflammation. The aging blood system becomes an evolving ecosystem. One clone expands not because it benefits the person, but because it competes well inside the marrow. Stem cells elsewhere face different pressures. Muscle satellite cells help repair skeletal muscle. Intestinal stem cells renew the gut lining. Skin stem cells replenish epidermis and hair follicles. Neural stem and progenitor cells persist in limited regions and roles. Stem cell exhaustion does not mean every tissue runs out of a fixed supply. Numbers, activation thresholds, niches, blood flow, inflammation, metabolism, and accumulated damage all change. A stem cell can remain present but become less responsive. Its surrounding niche can send the wrong signals. Repeated activation can deplete some populations. Dormancy protects stem cells from replication damage, yet excessive dormancy prevents repair. Again, the balance matters. Tissue aging becomes visible where these systems meet. Skeletal muscle loses mass and power with age, a process called sarcopenia when it becomes clinically significant. The decline involves motor neurons, muscle fibers, hormones, inflammation, protein synthesis, mitochondria, activity, nutrition, and satellite cells. Strength falls faster than muscle mass alone would predict. Power, the ability to produce force quickly, can decline even earlier. A person may still have enough muscle to stand under ordinary conditions, but lack the rapid response needed to prevent a fall. Muscle is also a metabolic organ. It stores glucose, releases signaling molecules during contraction, supports bone, and provides reserve during illness. Loss of muscle can make every stress harder to survive. Resistance exercise is one of the clearest ways to stimulate muscle protein synthesis and preserve strength in older adults. It does not stop aging. It changes the trajectory of one of its most consequential systems. Bone follows a different rhythm. Cells called osteoclasts remove mineralized tissue. Osteoblasts build new bone. Remodeling repairs microscopic damage and adapts the skeleton to load. In youth, formation and resorption support growth and strength. Later, resorption can exceed formation. Hormonal changes, especially the decline in estrogen around menopause, accelerate bone loss. Physical inactivity, low body weight, smoking, medications, disease, and inadequate nutrition can add risk. Bone density is only part of fracture resistance. Microarchitecture and quality matter. A fracture in old age is not simply a broken object. It can initiate immobility, muscle loss, blood clots, infection, isolation, and further frailty. One event exposes the interconnected system. Blood vessels age through changes in collagen, elastin, smooth muscle, endothelium, inflammation, and mineral deposition. Arteries become stiffer. The heart must pump against different pressure. Pulses travel more forcefully into small vessels in the brain and kidney. The endothelium, the thin cellular lining of blood vessels, produces signals that regulate dilation, clotting, and immune movement. Its function changes with age and cardiovascular risk factors. Atherosclerosis is not the inevitable presence of birthdays alone. It is a disease process involving lipoproteins, inflammation, vessel injury, genetics, blood pressure, smoking, diabetes, and time. Age increases opportunity for the process to accumulate. The heart itself changes. Cardiac muscle cells are largely long-lived. Some are lost and replaced poorly. The remaining cells enlarge. Connective tissue increases. The conduction system can become less stable. Maximum heart rate and reserve decline. At rest, function may appear adequate. During exertion or illness, reduced reserve becomes visible. The lungs lose some elastic recoil. The chest wall stiffens. Respiratory muscles can weaken. The surface area and vascular relationships involved in gas exchange change. A healthy older person can still breathe comfortably. A severe infection or surgery demands reserve that may no longer be present. The kidneys gradually lose filtering units in many people, though the rate varies widely. Blood flow and concentrating ability change. Medications remain in the body longer. Dehydration and electrolyte disturbances become easier. The liver retains remarkable regenerative ability, yet drug metabolism and blood flow can change. The skin becomes thinner in some layers and altered in others. Collagen organization changes. Wounds heal more slowly. Sweat and oil production shift. Sun exposure produces changes beyond intrinsic aging. The brain follows no single schedule. Some processing speed and aspects of memory change gradually. Vocabulary and accumulated knowledge can remain stable or improve for long periods. Brain volume changes differently across regions. White matter, blood vessels, synapses, myelin, inflammation, and protein clearance contribute. Normal cognitive aging is not dementia. Alzheimer's disease, vascular dementia, Lewy body disease, and other conditions involve specific pathologies whose risk rises with age. Age is the strongest risk factor for many neurodegenerative diseases because the brain has had more time to accumulate vulnerability and less reserve to contain it. But many very old people do not develop severe dementia. Aging changes probability, not destiny. The senses alter the entire organism's relationship with the world. The eye lens stiffens, making near focus harder. Retinal and neural changes affect contrast and low light vision. High-frequency hearing often declines as sensory cells and neural pathways are damaged. Smell can weaken. These changes influence balance, communication, nutrition, sleep, and social connection. An older body can become more isolated not only through circumstance, but because conversation is harder to hear and environments harder to navigate. Social biology enters cellular biology. Loneliness, chronic stress, poverty, unsafe housing, access to healthcare, education, and relationships all affect exposure, behavior, hormones, inflammation, and recovery. Aging is molecular without being only molecular. The body carries the conditions in which it has lived. This is why two people of the same chronological age can appear so different. One has accumulated disease, injury, stress, and environmental exposure. Another has genetic protection, resources, treatment, activity, and chance. Neither difference can be reduced to virtue. Healthy habits matter, but they operate within biology and circumstance. The concept of biological age arose from this variability. If chronological time does not fully describe function, perhaps measurements can estimate how far the organism has traveled along an aging trajectory. That possibility created clocks. It also created a new way for numbers to promise more than they know. A clock can measure time without explaining it. Chronological age is counted from birth. A biological clock attempts to estimate something else, the accumulated state of a tissue or organism. Researchers have built clocks from blood chemistry, physical performance, immune cell composition, brain imaging, protein levels, metabolites, gene expression, and DNA methylation. The most famous are epigenetic clocks. DNA methylation attaches small chemical groups to cytosine bases, often at sites where cytosine lies beside guanine. Patterns change with development, cell identity, environment, disease, and age. In 2013, Steve Horvath published a clock using methylation at hundreds of sites to estimate age across many human tissues. The prediction was remarkably accurate. A blood sample, skin sample, or organ could produce a molecular age close to the donor's chronological age. Later clocks were trained not only to predict birthdays, but to predict mortality risk, disease, or physiological decline. PhenoAge incorporated clinical risk relationships. GrimAge used methylation surrogates for proteins and smoking history to predict lifespan-related outcomes. Dunedin Pace was designed to estimate the pace of aging from longitudinal changes observed in a birth cohort. These tools changed research. An intervention could be tested over months or years without waiting decades for deaths. A disease could be associated with accelerated age. One tissue could be compared with another. But a clock is a model trained on a target. If it is trained to predict chronological age, it becomes good at recognizing patterns correlated with time. That does not mean every site causes aging. A gray hair can predict age without causing mortality. A clock trained on disease outcomes may predict risk better, but it still may not be a validated substitute for the outcome itself. Changing the clock does not automatically change health. An intervention might alter methylation at selected sites without improving function. A treatment could improve strength, immunity, or cardiovascular risk while leaving one clock unchanged. Different clocks can disagree about the same person. Technical platform, cell composition, sample handling, ancestry, disease, and algorithm influence results. This is why a biological age result should not be treated as a precise personal expiration date. The difference between predicted and chronological age can be statistically meaningful across groups and still noisy for one individual. Consumer tests often offer one number with more certainty than the science supports. The deeper scientific question is whether these clocks record damage, adaptation, or both. Some methylation changes may be harmful. Some may compensate for other changes. Some reflect shifting proportions of blood cells. Some may simply mark time. The clock face contains several mechanisms mixed together. Experiments that reverse epigenetic age therefore require caution. The most dramatic route begins with a discovery about cell identity. In 2006, Shinya Yamanaka and Kazutoshi Takahashi showed that a small set of transcription factors could convert mature mouse cells into induced pluripotent stem cells. The factors, commonly called OSKM after OCT4, SOX2, KLF4, and CMYC, reset much of the cell's epigenetic program. A skin cell could lose its specialized identity and return toward a state capable of becoming many tissues. The discovery transformed regenerative biology. It also posed a dangerous idea. If full reprogramming erases cellular age, could partial reprogramming make a cell younger without making it forget what it is? Full reprogramming inside a body would be hazardous. Cells that lose identity can form tumors called teratomas. Tissue organization could collapse. CMYC itself is associated with cancer. Partial reprogramming attempts to expose cells to selected factors briefly or cyclically. The aim is to reset some age-related epigenetic patterns while preserving identity. In mouse models of premature aging, cyclic expression of reprogramming factors improved selected tissues and extended survival. Other studies restored aspects of vision after optic nerve injury, altered muscle regeneration, reduced fibrosis, or shifted molecular age markers. Research published in 2025 reported that partial reprogramming could reverse a broad age-associated drift toward mesenchymal and fibrotic cell states in several models. These are important experiments. They are not evidence that a safe whole-body age reversal treatment exists for humans. Delivery remains difficult. Different tissues respond differently. Dose and duration must be controlled. Tumor risk, immune effects, loss of identity, and long-term stability must be understood. A cell may look younger by one epigenetic measure while retaining mutations, damaged proteins, mitochondrial changes, or altered tissue structure. Resetting instructions does not automatically replace every damaged part. The analogy is closer to restoring corrupted software while the hardware remains old. Even that analogy is incomplete because biological software and hardware continually create each other. Epigenetic information shapes repair. Repair reshapes epigenetics. This has led some researchers to describe aging as loss of information rather than accumulation of damage. The two ideas need not be opposites. Damage can disrupt regulation. Disrupted regulation can reduce repair. Aging can be a feedback loop in which information and material decline together. The promise of reprogramming is that some age-related states are not irreversible. The danger is assuming that reversibility in one assay equals rejuvenation of the whole organism. A clock can run backward while risk remains. Biological age also appears in physical tests. Walking speed predicts health outcomes in older adults because it integrates strength, balance, nervous function, heart, lungs, pain, vision, and confidence. Grip strength reflects muscle and neural function. The ability to rise from a chair combines power, joints, balance, and cognition. These tests are less molecular and often more directly connected with daily independence. Frailty indices count deficits across many systems. One small abnormality may matter little. Many small abnormalities together predict vulnerability. Frailty is not synonymous with old age. A young person with severe disease can be frail. An older person can remain robust. The index captures reserve across systems. This returns to Gompertz. Mortality rises because vulnerability accumulates, but the accumulation differs among people. No clock measures the whole process perfectly because aging has no single hidden variable. It is a pattern distributed across levels. This is also why apparent rejuvenation can be selective. A cataract operation can restore vision without changing immune age. A joint replacement can restore movement without lengthening telomeres. A vaccine can improve protection without reversing epigenetic clocks. Exercise can make muscle younger in function, while chronological age remains unchanged. Medicine has already reversed many individual consequences once accepted as inevitable. It treats blood pressure, replaces lenses, repairs arteries, prevents infections, controls diabetes, restores hearing, and rebuilds joints. These are not usually marketed as age reversal. They are examples of reducing the burden created by aging. The distinction between treating age-related disease and treating aging itself is partly conceptual. If several diseases share underlying mechanisms, targeting the mechanism could delay them together. This is the goal of geroscience. Rather than treating cardiovascular disease, cancer, dementia, diabetes, and frailty as entirely separate events, geroscience asks whether modifying aging biology can postpone several at once. The idea is reasonable. The proof requires clinical outcomes. A mouse living longer is evidence in a mouse. A biomarker changing is evidence about a biomarker. A drug preventing multiple human diseases would be stronger. A trial showing longer healthy life would be stronger still. The gap among these levels is where much of longevity marketing lies. A supplement increases NAD. A blood test reports a lower epigenetic age. A clinic calls the result rejuvenation. The scientific chain may stop before health or survival is demonstrated. Aging research needs enthusiasm because experiments are difficult and long. It also needs resistance to its own most attractive conclusions. The person being measured is more than the clock. Function, disease, cognition, independence, and lived experience matter more than one molecular age. The best clock would not merely tell how old a body appears. It would help predict which intervention preserves what the person can still do. That standard has not yet been fully reached. Nature offers another way to study biological time. Instead of asking how old one human tissue appears, researchers compare species that solved maintenance in radically different ways. Some bodies make the human lifespan look brief. Their stories begin in cold water, dark burrows, shells, and enormous hearts. The ocean quahog is not visually dramatic. It is a thick-shelled clam living in cold North Atlantic water, often buried partly in sediment. It feeds by filtering small particles and grows slowly. Its shell records annual bands. In 2006, researchers collected a large specimen near Iceland. By counting and cross-checking the growth lines, they estimated that the animal had lived for 507 years. It had begun growing around 1499. The clam was later nicknamed Ming because its life overlapped the Chinese Ming dynasty. The name made the specimen famous. Its biology is more important than the historical comparison. Arctica Icelandica can maintain proteins, membranes, and cells across centuries. Cold water and slow metabolism likely reduce some forms of damage, but temperature alone cannot explain everything. Closely related species living in similar environments do not always live as long. The clam's proteins show unusual stability. Antioxidant and stress responses remain effective. Mitochondria appear to produce relatively little damaging leakage under some conditions. Cell death pathways are regulated in ways that may preserve tissue. The longest-lived individual does not reveal one immortality gene. It reveals a suite of maintenance traits working together at a slow pace. A shell also makes age easier to verify. Human claims beyond 100 years depend on documents that can be missing or incorrect. A clam carries its chronology in layers. The Greenland shark carries time in a part of the eye. The shark lives in cold North Atlantic and Arctic waters. It moves slowly, grows slowly, and can reach several meters in length. Ordinary fish aging methods often use hard structures that add growth rings. Greenland sharks lack the appropriate calcified tissues. Researchers turn to the lens of the eye. Proteins in the center of the lens form before birth and remain metabolically inert. They preserve a chemical signature from the animal's early life. A 2016 study used radiocarbon dating of eye lens nuclei from 28 female sharks. The two largest animals produced age estimates spanning several centuries. The analysis supported a lifespan of at least about 272 years, with the largest specimen's central estimate near 4 centuries and a wide uncertainty range. Popular retellings converted the upper end of that uncertainty into a precise claim that one shark was 512 years old. The science does not provide that exact birthday. Radiocarbon dating at such ages depends on calibration, marine carbon sources, growth assumptions, and the nuclear bomb pulse visible in younger animals. The secure conclusion is already extraordinary. Greenland sharks are probably the longest-lived known vertebrates, and some may live for several centuries. They may not reach sexual maturity until roughly a century or more has passed. A shark born before industrialization could still be moving through cold water today. Its long life is not a model that can simply be transferred to humans. The animal's body temperature is near freezing. Metabolism is slow. Growth is measured in centimeters across years. Reproduction begins late. Its tissues evolved under conditions no human organ experiences. Yet its genome and physiology can reveal which repair and stability systems become important when a vertebrate body must endure for centuries. Researchers have investigated DNA repair, immune genes, chromatin, iron metabolism, and protein maintenance. The answer again appears distributed. There is no shark molecule that stops time. The bowhead whale presents the opposite problem of scale. A whale contains vastly more cells than a mouse. More cells mean more opportunities for mutations that could initiate cancer. A longer life adds more time for those mutations to accumulate. If risk rose simply with cell number and lifespan, large long-lived animals should develop cancer constantly. They do not. This mismatch is called Peto's paradox. Across species, cancer risk does not increase in direct proportion to body size. Large animals have evolved additional suppression. Elephants carry extra copies of genes involved in DNA damage responses, including forms related to TP53. Bowhead whales, which can live beyond 2 centuries, show distinctive changes in genes related to DNA repair, cell cycle regulation, and metabolism. Recent experiments have suggested that bowhead whale cells may repair certain double-strand DNA breaks with unusually high accuracy. Different large species solve the problem differently. Evolution cannot build a huge body merely by multiplying ordinary cells without changing cancer control. Longevity and body size force improvements in surveillance, repair, tissue architecture, or elimination of damaged cells. The whale's slow life contains another historical clue. Inuit hunters have found old stone or metal weapon fragments embedded in bowhead tissue. Some designs belong to hunting technologies used more than a century earlier. The healed tissue had carried the fragment through decades. A wound became a timestamp inside a living animal. Naked mole rats solve longevity in a body closer to a laboratory mouse. They are small rodents that live underground in East Africa. Their skin is loose, their vision limited, and their colonies are organized around one breeding female and a small number of breeding males. The burrows can be crowded, low in oxygen, and high in carbon dioxide. A mouse of similar size may live several years. A naked mole rat can live beyond 30. Early demographic analysis found no statistically significant rise in mortality with age across much of the available captive data, an apparent violation of the usual Gompertz pattern. The phrase negligible senescence became attached to the species. It should not be interpreted as proof that the animals never age. Very old naked mole rats can develop disease, functional decline, and cancer. Population data at extreme ages remain limited because few individuals reach them. Later work continues refining whether mortality truly remains flat. The important point is relative. The animals age remarkably slowly for their size. Their proteins resist unfolding and aggregation. Proteasome activity and autophagy remain effective. Ribosomes appear unusually accurate in protein synthesis. Cells tolerate low oxygen. Inflammatory and DNA damage responses differ from those of mice. Their tissues contain very high molecular weight hyaluronan, a long sugar polymer in the extracellular matrix. This material contributes to the unusual elasticity of their skin and has been linked with strong contact inhibition, a mechanism that stops cells dividing when crowded. Experiments altering hyaluronan pathways reduced some cancer resistance in cultured cells. The molecule is part of the explanation, not all of it. In 2025, researchers reported another mechanism involving C-gas, an immune sensor that detects DNA in the cell's cytoplasm. In many mammals, C-gas activation is associated with inflammation and can interfere with aspects of DNA repair. Naked mole rat C-gas showed differences that appeared to support genome stability and resistance to age-related decline. One protein had evolved a different relationship with damage. The discovery does not mean a naked mole rat gene can be added safely to a person. It shows that pathways familiar in humans have alternative configurations. Evolution found another setting. Some animals challenge the definition of aging itself. Hydra are tiny freshwater animals with tubular bodies and tentacles. They maintain populations of stem cells that continually renew tissues. Under protected laboratory conditions, some hydra populations show little or no increase in mortality or decline in reproduction across the observed interval. They can still die from infection, starvation, temperature, or injury. They are not indestructible. Their body organization allows continuous replacement in a way that a human brain or heart cannot copy. Planarian flatworms can regenerate large portions of the body from small fragments. Their stem cells retain extraordinary developmental potential. Some jellyfish can return from a mature form to an earlier life stage under stress, creating a cycle that is often described as biological immortality. The term exaggerates. The animals can be eaten, diseased, or damaged. The reversal is not guaranteed. Their nervous systems and tissue organization differ profoundly from those of vertebrates. Negligible senescence does not mean invulnerability. It means that the internal risk associated with age rises slowly or remains difficult to detect. Turtles and tortoises add another pattern. Several species live for more than a century. Studies across zoos and wild populations show that some chelonians have very slow demographic aging, while others still show measurable senescence. Protected bodies, slow life histories, and flexible metabolism contribute. Temperature and environment influence their rates. The word reptile does not imply one metabolism or lifespan, just as mammal does not. Species comparisons reveal broad trends. Larger animals often live longer within a related group, partly because their metabolism per gram is lower and predation risk differs. Flying animals often outlive non-flying animals of similar size. Animals protected by shells, size, venom, social systems, or inaccessible habitats can evolve slower aging because more individuals survive long enough for late maintenance to matter. But exceptions are as informative as trends. Bats maintain high metabolic activity and still live long. Birds have high body temperatures and can be long-lived. Naked mole rats tolerate oxidative damage without simply eliminating it. Long-lived species do not all minimize the same molecule. They preserve function through different combinations of repair, resistance, turnover, cancer suppression, and ecology. This limits the search for one universal longevity mechanism. A bowhead whale cannot use the same strategy as Hydra. A neuron lasting a century cannot be maintained exactly like an intestinal cell replaced every few days. Even within one human body, tissues need different solutions. The most useful lesson from long-lived animals is not that slow metabolism guarantees long life. It is that aging rate is biologically adjustable. Nature has altered it repeatedly. The human rate is not the only rate possible for living matter. At the same time, evolutionary distance matters. A clam can keep simple tissues for 5 centuries without preserving a human-like brain, upright skeleton, immune history, and reproductive system. A Greenland shark trades speed and early reproduction for endurance. A naked mole rat lives within a colony and burrow environment unlike human life. Longevity is attached to a whole organism and its history. Borrowing one mechanism may help a disease. It may not recreate the life history that made the mechanism valuable. Human longevity must therefore be studied in humans. The oldest human lives are shorter than those of sharks and clams, but they carry a different kind of evidence. They show how genes, public health, family, wealth, chance, disease, and documentation combine at the edge of one species' survival. For most of human history, average life was short. This does not mean everyone died at 30. High infant and childhood mortality pulled averages downward. People who survived early infection, injury, childbirth, famine, and violence could reach old age. Still, far fewer did. Clean water, sanitation, vaccination, antibiotics, safer childbirth, food security, housing, education, and treatment of cardiovascular disease transformed survival. Life expectancy rose not because human cells suddenly began aging differently, but because deaths that once occurred before old age were prevented. This distinction separates lifespan from aging rate. A population can live longer because it reaches aging more often. Later improvements can also delay disease within old age, but public health remains one of the greatest longevity interventions ever achieved. The record at the far end is held by Jean Calment. She was born in Als, France, in 1875 and died there in 1997 at the validated age of 122 years and 164 days. Her life crossed from the era before electric lighting was common into the age of the internet. Calment became famous for her memory, humor, and stories, including acclaimed childhood encounter with Vincent van Gogh. Extreme ages require more than a story. Demographers inspect birth records, censuses, marriage documents, family relationships, and continuous identity across life. At very advanced ages, one mistaken birth certificate, name reuse, or family substitution can create a false record. Calment's documentation has been challenged, most dramatically by a hypothesis that her daughter assumed her identity. Detailed reviews by specialists in age validation have continued to support the original record, while acknowledging why extreme claims deserve unusual scrutiny. No independently validated person has surpassed her age. Her record does not prove that 122 is a fixed biological wall. A rare maximum depends on population size and chance. If more people reach 110, the probability that one reaches 120 increases even if individual biology does not change. Statistical studies disagree about whether human lifespan has a hard limit, a soft practical ceiling, or no detectable fixed boundary within current data. Mortality appears to decelerate at the most extreme ages in some datasets, with annual risk approaching a very high plateau. Other analyses find continuing increases or emphasize uncertainty caused by small samples and age errors. After 110, the population becomes tiny. One incorrect date can distort the curve. The safest conclusion is narrower than either immortality or a precise ceiling. Human survival beyond 115 is rare. Survival beyond 120 is extraordinarily rare. No existing evidence shows that current medicine can produce routine lifespans far beyond the validated record. The increase in life expectancy during the last century has not been matched by a comparable march in maximum verified age. More people approach the frontier. The frontier has moved little. Centenarians provide a larger group for study. People reaching 100 are not all healthy in the same way. Some avoid major disease until late. Some survive disease for many years. Some remain cognitively strong while physically frail. Others preserve mobility but develop dementia. Researchers sometimes distinguish escapers, delayers, and survivors. Escapers reach very old age without major common diseases until near the end. Delayers develop them later than average. Survivors develop disease at more ordinary ages but live with it for a long time. Longevity is therefore not one phenotype. Genes contribute. Variants near APOE influence lipid transport and Alzheimer's disease risk. APOE4 is less common among many centenarian groups, while E2 can be more common. Variants in FOXO3 have been associated repeatedly with exceptional longevity across several populations. FOXO proteins respond to insulin signaling, stress, metabolism, and repair. Other associations involve immune function, cholesterol, DNA repair, and inflammation. No longevity gene guarantees a long life. Common variants have small effects. Different populations carry different combinations. A protective variant for one disease may increase another risk. For many years, twin and pedigree studies placed the heritability of human lifespan broadly around 6 to 25%, with genetic influence appearing stronger at exceptional ages. A 2026 analysis in Science challenged that familiar range. The researchers argued that deaths from infections, accidents, violence, and other external causes obscure the inherited component of intrinsic lifespan. After modeling those competing causes and reanalyzing twin cohorts, they estimated that intrinsic human lifespan could be more than 50% heritable. The result is important and not a final division of every life into fixed genetic and environmental shares. Heritability describes variation within particular populations and conditions. It does not say that half of one person's years are predetermined by genes. Changes in sanitation, smoking, medicine, inequality, and exposure can alter both lifespan and the estimated proportion attributed to inheritance. The new estimate also depends on how intrinsic and extrinsic mortality are separated, and other researchers continue debating the assumptions. The cautious conclusion is that genetic influence on human lifespan may be substantially larger than older estimates suggested, while environment, behavior, healthcare, social conditions, and chance remain powerful. This makes centenarian families scientifically valuable without turning their genomes into destiny. Children and siblings of centenarians often show delayed cardiovascular disease or favorable metabolic profiles. They also may share food, wealth, location, habits, and healthcare. Separating inheritance from environment requires large cohorts and careful comparison. Sex creates another strong pattern. Women outlive men in nearly every modern population. The gap changes across countries and eras, showing that biology is not the only cause. Men historically experienced more smoking, hazardous work, violence, and risky behavior in many settings. Women face pregnancy and childbirth risks where healthcare is limited, yet still often survive longer later. Sex chromosomes, hormones, immune responses, fat distribution, and cardiovascular biology contribute. 2X chromosomes may provide cellular mosaic protection against some harmful variants. Estrogen influences vessels and metabolism before menopause. Testosterone, body size, and immune differences may alter risk. No single explanation accounts for the gap. Longevity is produced by biology inside society. This becomes clear when life expectancy is mapped by income, education, race, neighborhood, occupation, and access to care. People living only a few kilometers apart can differ by years in expected lifespan. Air quality, heat, housing, food, chronic stress, medical treatment, violence, and work enter the body through different mechanisms. Aging research that focuses only on molecules can mistake unequal exposure for individual biology. A damaged artery does not record whether inflammation came from a gene, pollution, smoking, stress, infection, or several together. The tissue contains the result. Reproductive aging follows another clock. In ovaries, the number of immature egg-containing follicles is established largely before birth and declines across life. Most follicles never release an egg. They are lost through a process called atresia, while a much smaller number reach ovulation. The decline is not a simple countdown with one identical rate for every person. Genes, smoking, medical treatment, surgery, immune disease, and other factors can shift timing. As the remaining follicle pool falls and hormonal signaling becomes more variable, menstrual cycles change. Eventually, ovarian production of estrogen and progesterone changes enough that menstruation stops. Menopause is defined retrospectively after 12 months without a menstrual period when no other cause explains it. It usually occurs years before the end of the human lifespan. This makes humans unusual. Most female mammals remain capable of reproduction close to death, though reproductive decline still occurs. Clear prolonged post-reproductive life has evolved in humans and a small number of toothed whales, including killer whales. Why would evolution support survival after reproduction ends? One explanation is the grandmother hypothesis. Older females can increase the survival of descendants through food, care, ecological knowledge, and support of daughters and grandchildren. In killer whales, post-reproductive females can lead groups towards salmon and become especially valuable when food is scarce. Human evidence is more complex because culture, kinship, male reproduction, birth risk, and long childhood all interact. Menopause may not have evolved for one reason. It can arise from the relationship between ovarian biology, maternal mortality, intergenerational support, and the unusual human life history. The reproductive clock also demonstrates why aging cannot be summarized by one biological age. A person can enter menopause while muscle, cognition, cardiovascular reserve, and immune function remain strong. The transition changes bone remodeling, temperature regulation, sleep, blood vessels, urogenital tissue, and metabolism through altered hormones. It does not switch the entire organism from young to old. Symptoms vary widely. Some people experience hot flashes, disrupted sleep, mood changes, or pain. Others have few difficulties. Medical treatment can be appropriate depending on symptoms, health history, age, and individual risk. Menopause is a normal life stage and also a biological transition with treatable consequences. Both descriptions can be true. Male reproductive aging is generally more gradual. Sperm production can continue into advanced age, but hormone levels, semen quality, erectile function, and mutation rates in sperm change. Older paternal age is associated with higher risk for selected new mutations and some outcomes in offspring, though absolute risks for any one pregnancy can remain small. Reproduction, therefore, does not divide humans into one sex that ages and another that does not. The schedules differ. The reproductive system also participates in the trade-off between regeneration and cancer. Estrogen supports bone and several tissues, yet lifetime exposure can influence the risk of hormone-sensitive cancers. Testosterone supports muscle, bone, and sexual function, while treatment can affect prostate, blood, and cardiovascular systems depending on context. Replacing a hormone is not equivalent to restoring youth. It changes signaling within an older body. This principle extends to growth hormone. Levels fall across adulthood, and deficiency caused by pituitary disease can be treated medically. Giving growth hormone to healthy older adults can increase lean mass modestly while causing swelling, joint pain, insulin resistance, and other harms. It has not been shown to extend life. A youthful level of one hormone is not always optimal later. The body changes receptor sensitivity, tissue composition, and risk. Aging is not the loss of a single youthful mixture that can be poured back in. It is a new biological context. The long post-reproductive period also changes the meaning of health span. Preserving bone, cardiovascular health, cognition, continence, sexual comfort, and social role becomes more important than preserving fertility indefinitely. A species that depends on learning and cooperation can benefit from bodies that remain useful beyond reproduction. Human longevity is therefore not only the persistence of cells, it is the persistence of participation. The idea of blue zones tried to learn from places with unusual concentrations of very old people. The term first arose in demographic work identifying a region of Sardinia with high male longevity. Researchers marked the area on a map in blue. It later expanded through popular books and media to include Okinawa, Ikaria, the Nicoya Peninsula, and the Seventh-day Adventist community in Loma Linda. The popular pattern emphasized ordinary movement, plant-rich diets, social connection, purpose, family, modest energy intake, and low smoking. Many of these factors are supported independently by public health evidence. The Blue Zone package is more complicated. The regions were identified through different methods and time periods. Some have changed rapidly. Okinawa's younger generations no longer show the same health profile as earlier cohorts. Migration and population denominators affect rates. Birth records vary in quality. Critics have argued that some extreme age clusters can arise from poor documentation, age exaggeration, pension fraud, or selective storytelling. Demographers involved in validating blue zones have responded that the core regions were checked through multiple records and established methods. The dispute is not resolved by treating every longevity claim as fraud or every branded lesson as proven. Some populations do show unusual survival. Their causes cannot be reduced to 9 lifestyle slogans: genetics, historical diet, infection, physical terrain, family structure, medical access, migration, and who remained in the region all matter. Commercial branding can turn a demographic observation into a recipe. Science must separate the observation from the product. One lesson survives the debate. Longevity is often embedded in daily environment rather than one heroic intervention. Walking occurs because destinations are nearby. Food patterns reflect agriculture and culture. Social contact is built into family and community. Smoking prevalence is shaped by policy. Healthcare and inequality matter. A person does not make every health decision in isolation. The environment repeatedly makes some choices easier than others. This helps explain why lifestyle research is difficult. People who exercise often differ from people who do not in income, disease, diet, medical care, and earlier health. People may stop exercising because illness is already developing, making inactivity appear more causal than it is. Randomized trials can test particular interventions, but rarely last a lifetime. Observational cohorts provide decades of data, but contain confounding. The strongest conclusions emerge when different methods agree. Smoking shortens life and increases disease through overwhelming converging evidence. Physical activity reduces risk across randomized trials of function and long observational studies of disease and mortality. Blood pressure control prevents cardiovascular events. Vaccination prevents infection. Treating high cholesterol in appropriate risk groups reduces heart attack and stroke. These interventions do not require a theory of immortality. They preserve systems known to fail. Diet is harder to compress. Food is a mixture of nutrients, culture, energy, and substitution. Eating more of one item usually means eating less of another. Patterns rich in minimally processed plant foods, fiber, unsaturated fats, and adequate protein are associated with better cardiometabolic outcomes in many populations. Excess energy, tobacco, heavy alcohol use, and highly processed diets can increase risk. But no single food has been shown to produce exceptional lifespan, and nutritional needs change with age, disease, medication, and body composition. In older adults, preventing undernutrition and muscle loss can be more important than imitating chronic calorie restriction. The diet that supports a young laboratory mouse is not a universal prescription for an 80-year-old person recovering from illness. Longevity science becomes most humane when it measures health span. Health span is the period of life spent with function, independence, and acceptable health. It is not the absence of every diagnosis. A person can live well with controlled hypertension, a replaced joint, hearing aids, or medication. The goal is to compress severe disability and disease toward a shorter period near the end of life. This idea is called compression of morbidity. If lifespan rises while years of disability rise equally, survival has improved without full healthy aging. If disease begins later and the period of frailty shortens, both quantity and quality change. Human longevity is therefore not only the age written at death, it is the shape of the years before it. The record holder attracts attention because 122 is a clear number. Health span is quieter. It appears as stairs still climbed, conversations still heard, meals still prepared, memories still accessible, and recovery still possible. The biology behind those abilities can now lead into intervention. Some interventions are already proven to preserve function. Others remain experiments. The difficulty is keeping the difference visible. The strongest interventions in human aging are not the most exotic. They are powerful precisely because they affect several systems at once, and because their outcomes have been measured in people. Physical activity is the clearest example. Movement places controlled stress on the body. A muscle fiber experiences tension. Energy levels fall temporarily. Calcium signals change. Mechanical force travels through connective tissue and bone. The heart pumps more. Blood vessels widen. Temperature rises. The body interprets the disturbance as a reason to adapt. Mitochondria increase in number and capacity. Capillaries grow. Muscle fibers add contractile proteins. Insulin sensitivity improves. Bone responds to load. Motor circuits become more efficient. Anti-inflammatory signals are released. The benefit does not come from avoiding stress. It comes from recovering from a stress small enough to stimulate repair. This phenomenon is often called hormesis. A low or moderate challenge activates protective systems. An overwhelming challenge causes damage. Exercise is one form. Heat, cold, fasting, and plant chemicals are sometimes placed under the same label, though evidence differs greatly among them. The word hormesis can become a justification for any uncomfortable practice. A biological response to stress does not guarantee that more stress extends life. Dose and recovery remain central. Aerobic exercise increases the body's ability to transport and use oxygen. Cardiorespiratory fitness predicts cardiovascular disease and mortality strongly across populations. Improvement can occur even when body weight changes little. Walking, cycling, swimming, running, and many other activities can train the system. The exact activity matters less than repeated demand appropriate to the person's capacity. Resistance exercise protects another form of reserve. Muscle contracts against load. Protein synthesis rises during recovery. Motor units are recruited and refined. Tendons and bones adapt. In later life, resistance training can increase strength even when begun after decades of inactivity. The older body retains plasticity. It may need more recovery and careful progression, but it is not unable to respond. Balance practice and power training address rapid control. A fall occurs in fractions of a second. The nervous system must detect movement, produce force, and place a foot before conscious planning is complete. Strength without speed may not be enough. Exercise also reaches the brain. Blood flow changes. Growth factors including B, D, and F are influenced. Sleep and mood can improve. The hippocampus and connected networks respond to activity. The claim that exercise creates large numbers of new human neurons is stronger than current evidence allows. But exercise supports vascular, metabolic, and synaptic conditions associated with cognitive health. No pill reproduces the complete signal of moving a body through space. Exercise has risks and limits. Excess training without recovery can cause injury, hormonal disturbance, and immune stress. Frailty, heart disease, pain, or disability require adaptation. Movement cannot prevent every dementia, cancer, or genetic disease. A strong association with longevity is not a guarantee for one person. Still, compared with experimental longevity drugs, physical activity has unusually broad human evidence for extending health span and reducing premature death. Sleep is another system-wide behavior, though its relationship with aging is bidirectional. Sleep changes across the lifespan. Deep slow-wave sleep is abundant in childhood and tends to decline with age. Circadian timing can shift earlier. Sleep becomes more fragmented. Medical conditions, pain, medication, menopause, prostate symptoms, breathing disorders, and changes in daily activity contribute. Older adults do not simply need no sleep because they obtain less. Chronic short or disrupted sleep is associated with cardiovascular disease, metabolic dysfunction, impaired immunity, depression, accidents, and cognitive decline. Association does not always establish direction. Disease can disturb sleep before diagnosis. Sleep apnea can produce both fragmentation and cardiovascular stress. Pain can keep a person awake, while poor sleep increases pain sensitivity. The relationship forms a loop. During sleep, the nervous system changes state. Hormones follow rhythms. Immune responses are coordinated. Memory is processed. Metabolism and appetite regulation change. Growth hormone release is linked strongly with early deep sleep, especially in younger people, but repair does not occur only during sleep or through one hormone. The popular statement that the brain washes out toxins every night is too simple. Sleep alters cerebrospinal fluid dynamics, neural activity, blood flow, and molecular clearance. Animal and human studies support links between sleep and removal of some metabolites, while experiments disagree about exact mechanisms and rates. Sleep is not a nightly rinse that can erase daytime aging. It is one part of integrated regulation. Trying to force perfect sleep can create its own vigilance. The relevant biological pattern is regular opportunity, circadian alignment, breathing that remains unobstructed, and treatment of disorders when present. Aging can make sleep more difficult, and disturbed sleep can make aging-related vulnerability worse. Nutrition interacts with both movement and sleep. Protein provides amino acids for muscle, enzymes, antibodies, and tissue repair. Young bodies respond strongly to small protein doses. Older muscle can become less sensitive, a phenomenon called anabolic resistance. A larger or better distributed protein stimulus may be needed to produce the same synthesis response, especially alongside resistance exercise. This complicates simple longevity messages about reducing growth signaling. Chronic overactivation of nutrient pathways may promote aging mechanisms in animals. Insufficient protein in an older human can accelerate frailty. The best molecular state in a yeast cell is not automatically the best diet for preserving a person's ability to stand. Fiber supports bowel function and feeds microbial communities that produce metabolites affecting the intestine and immune system. Unsaturated fats, whole grains, legumes, vegetables, fruit, and minimally processed foods form patterns associated with lower cardiometabolic risk. Adequate energy prevents wasting. The body needs enough material to maintain itself. The field of longevity sometimes treats hunger as evidence that a pathway is working. Human health does not require proving discipline through deprivation. Smoking provides a clearer case because it accelerates several hallmarks at once. Tobacco smoke damages DNA, injures blood vessels, promotes inflammation, alters immune function, shortens telomeres in population studies, and causes cancer, lung disease, stroke, and heart disease. Stopping reduces risk at any age, though some damage persists. The intervention does not target an abstract aging pathway. It removes a repeated source of injury. Air pollution acts more diffusely through particles and gases that reach lungs, vessels, and inflammatory systems. Individual behavior cannot eliminate exposure created by cities, industry, housing, or work. Longevity is partly environmental policy. Alcohol illustrates changing evidence and cultural habit. For years, moderate drinking was portrayed as protective, partly because observational studies found lower cardiovascular risk among moderate drinkers than abstainers. The abstainer group often included former heavy drinkers or people who stopped because of illness. Social and economic differences also confounded results. More rigorous analyses have weakened the idea of a health-promoting dose. Alcohol increases risk for several cancers and other diseases. Any possible cardiovascular trade-off does not make it an anti-aging treatment. A traditional practice in a long-lived region is not proof of causation. Social connection affects health through pathways less visible than a drug receptor. Isolation can alter stress, sleep, activity, medical care, nutrition, and immune regulation. A person living alone may have no one to notice infection, a fall, confusion, or weight loss. Relationships can provide practical help and biological safety. They can also be sources of chronic stress. The number of contacts matters less than their quality and reliability. A sense of purpose is difficult to randomize in a trial, but goals and social roles influence behavior, cognition, and recovery. The body ages inside a social world. Preventive medicine changes the trajectory without changing the calendar. Blood pressure treatment reduces stroke and heart failure. Statins reduce cardiovascular events for people whose risk justifies them. Vaccination prevents infections that become more dangerous with age. Cancer screening can detect selected diseases earlier. Hearing correction can improve communication and reduce isolation. Vision treatment prevents falls and restores function. These are interventions against age-related vulnerability. None makes a cell 20 years younger. Together, they can add healthy years. This raises a regulatory question. Must a treatment target one disease, or can it target the shared biology that makes many diseases more likely? The answer matters for experimental geroprotective drugs. Clinical trials need endpoints. A lifespan trial in healthy middle-aged adults could take decades. Biomarkers are faster but may mislead. A composite of cardiovascular disease, cancer, dementia, and death may reveal whether several conditions are delayed together. Metformin became the proposed test case. The drug has been used for decades in type 2 diabetes. It reduces liver glucose production and affects cellular energy pathways, including AMPK, though its actions are broader and not fully resolved. Observational studies sometimes found lower rates of cancer or mortality among people with diabetes taking metformin than among comparison groups. These studies contain major confounding. People prescribed different drugs differ in disease severity, kidney function, weight, medical care, and survival to treatment. Metformin extends lifespan in some animal models and not others. Its effects can depend on dose, sex, diet, genetics, and whether the organism has metabolic disease. The Targeting Aging with Metformin trial, known as TAME, was designed as a 6-year, multi-center test of whether metformin could delay a composite of age-related diseases in older adults without diabetes. Its importance was partly regulatory. A successful trial could show that aging-related risk is a modifiable target across diseases. As of mid-2026, TAME remained an upcoming trial rather than a completed test with efficacy results. Its organizers were still describing the project as a study to be launched, so it could not yet demonstrate that metformin slows human aging or extends health span. The proposal remains influential. The drug is not established as a longevity treatment for healthy people. Metformin can cause gastrointestinal effects, vitamin B12 deficiency, and rare serious complications in susceptible patients. It may also alter some adaptations to exercise in certain studies. A medication beneficial for diabetes does not become harmless when taken for a speculative purpose. The proven and experimental interventions now stand beside one another. Exercise changes outcomes in people. Blood pressure treatment changes outcomes in people. Rapamycin changes lifespan in mice and selected markers in small human studies. Metformin has extensive disease use and incomplete geroscience evidence. Senolytics remove cells in animals and are only beginning human testing. NAD precursors raise metabolites more reliably than they improve function. Partial reprogramming changes old tissues in experimental models and carries profound risks. The hierarchy should remain visible. Possibility is not proof. Aging research becomes more trustworthy when it can say both that biology is modifiable and that no current intervention has made normal human aging disappear. The experimental frontier is shaped by a simple question. Can a mechanism that contributes to aging be altered without disrupting the useful work it performs? Senolytics offer one test. In laboratory mice, eliminating selected senescent cells can improve cardiac function, physical endurance, bone, fat tissue, and other age-related outcomes in some models. The effect is often strongest when senescent burden has been created by disease, radiation, or accelerated aging. Human senescent cells are more diverse than one laboratory target. A skin cell made senescent by ultraviolet light differs from a liver cell after injury or an immune cell after repeated activation. No universal marker identifies every harmful senescent cell and excludes every useful one. Dasatinib, quercetin, fisetin, navitoclax, and other compounds have been investigated for senolytic activity. Several were developed for other purposes and have significant effects outside senescent cells. Navitoclax can reduce platelets. Dasatinib is a cancer drug. Quercetin and fisetin are sold as supplements, but the doses and pharmacology used in experiments do not make ordinary dietary products proven senolytics. Early clinical studies have tested intermittent dosing in conditions including pulmonary fibrosis, kidney disease, diabetes-related dysfunction, and Alzheimer's disease. Some report changes in physical measures, inflammatory markers, or senescence-associated signals. Sample sizes are small. Control groups and blinding vary. Disease-specific results cannot be generalized to healthy aging. The central human question remains unanswered. Will clearing selected senescent cells improve meaningful function without causing delayed harm? Xenomorphic drugs take a gentler approach. They attempt to suppress the inflammatory secretions of senescent cells while leaving the cells in place. Rapamycin and related pathways can alter the secretory phenotype. Other compounds target NF-kappa B, P38 MAP kinase, or inflammatory cytokines. Reducing one harmful signal may preserve beneficial roles. It may also leave damaged cells occupying tissue. The best approach may differ by organ and stage. Immune rejuvenation is another frontier. The aging immune system contains fewer naive cells, persistent inflammatory clones, and altered bone marrow output. Researchers have explored thymic regeneration, vaccines designed for older immunity, cytokine modulation, removal of dysfunctional clones, and engineered immune cells. A small uncontrolled study combining growth hormone, metformin, and other agents reported changes in thymic imaging and epigenetic age. The design could not establish which treatment caused what, and growth hormone carries metabolic and cancer-related concerns. An intriguing result becomes a hypothesis, not a protocol. Vaccination already offers a practical form of immune support. It does not make the immune system young. It provides advanced information about specific threats, reducing the demand placed on an older response. The distinction between rejuvenation and compensation is often less important to the person protected. Stem cells attract even stronger claims. Bone marrow transplantation and blood stem cell therapies are established treatments for specific cancers and disorders. Cultured skin and corneal stem cells can repair selected tissues. Mesenchymal stromal cells are investigated in many diseases because they release signals and influence inflammation. Commercial clinics often advertise proven stem cell injections for general aging, joint pain, neurological disease, or fatigue. Injected cells may fail to survive, become trapped in organs, trigger immune reactions, transmit contamination, or form abnormal tissue. The phrase stem cell describes a property, not one medicine. A treatment must specify the cell source, identity, manufacturing, dose, route, target, and clinical evidence. Youthful cells placed into an old environment may adopt old behavior. An old niche can alter a young stem cell. Repairing the seed without changing the soil may be insufficient. Experiments joining the circulation of young and old animals made this interaction vivid. In heterochronic parabiosis, a young and old mouse are surgically connected so that they share blood. Early studies found changes in muscle, liver, brain, and other tissues of the older animal. The results were popularly described as young blood reversing aging. The experiment changes much more than blood. The animals share circulation, hormones, immune cells, metabolites, and the function of organs that process those molecules. The young animal can remove or dilute factors from the old animal. The old animal can expose the young one to harmful signals. Later experiments using plasma exchange and controlled factors suggested that reducing age-elevated molecules may be as important as adding youthful ones. No identified cocktail has been shown to reverse whole-body human aging. Commercial young plasma infusions appeared before evidence and prompted regulatory warnings. Plasma is a medical product with risks of allergic reaction, infection, clotting disturbance, and volume overload. A striking mouse image became a human marketplace without the necessary bridge. The same pattern appears in gene therapy. A gene can increase lifespan in mice under one controlled condition. Delivering it safely to billions of human cells is another problem. Telomerase gene therapy has extended health or lifespan in some mouse studies. Excess or poorly controlled activity could promote cancer. FOXO, autophagy, DNA repair, and mitochondrial genes are possible targets. Each participates in multiple tissues and developmental roles. A pathway selected for a long-lived species exists within that species' whole regulatory network. Increasing one component in humans can create imbalance. Partial epigenetic reprogramming may eventually become more controllable than permanent gene addition. Researchers are testing alternative factor combinations, transient RNA, proteins, small molecules, and tissue-specific delivery. The goal is to move cells toward youthful regulation without crossing into pluripotency. As of 2026, the most persuasive evidence remains preclinical. No approved therapy has safely rejuvenated the entire human body through reprogramming. The line between research and self-experimentation can become dangerous because aging affects everyone and progresses slowly. A person taking an unproven intervention may feel well for years whether the treatment works or not. There is no immediate signal, like a tumor shrinking or an infection clearing. Biomarkers can fluctuate. Expectations change perception. People using expensive treatments often change diet, exercise, sleep, and medical care at the same time. Without controls, the treatment receives credit for the whole package. Long-term harms may appear after the promoter has moved to another product. Longevity trials face unusual design problems. Participants may take several supplements secretly. They may guess their group from side effects. Outcomes occur slowly. A trial can measure a molecular change without knowing whether it is helpful. The most convincing programs use randomized designs, meaningful functional endpoints, independent replication, and enough follow-up to detect harm. The field also needs diverse participants. Many aging studies historically enrolled limited ancestry, socioeconomic groups, or one sex. A clock trained in one population may perform differently in another. Drug metabolism, disease risk, diet, and exposure vary. A universal anti-aging claim built on a narrow sample can widen inequality. Access creates a broader ethical question. If a treatment delays disease but is expensive, who receives the added healthy years? Longevity is already unequal. Wealthier populations often live longer because they encounter less hazard and more care. A technology that benefits only the wealthy could expand the gap, even if it works biologically. Longer life also changes pensions, caregiving, work, housing, and relationships across generations. These are not reasons to avoid research. They are part of what successful research would change. The language of war is common in longevity. Defeat aging. Escape death. Treat the body as an enemy. Biology offers a quieter frame. Aging is not an invader. It is the long-term behavior of systems that grew, reproduced, repaired, adapted, and survived through trade-offs. Reducing suffering is a medical goal. Preserving cognition, movement, immunity, and independence is a medical goal. Calling every visible sign of age a failure can turn treatment into rejection of older people themselves. A wrinkle is not the same as dementia. Gray hair is not frailty. Menopause is not a disease, though its symptoms and health consequences may need care. The value of a life is not measured by a youthful biomarker. Longevity science works best when it distinguishes appearance from function and function from worth. The future may produce combinations rather than one cure. A senolytic for one tissue. A vaccine adapted to older immunity. A drug that modulates mTOR intermittently. A therapy that restores a stem cell niche. A reprogramming pulse delivered locally after injury. Better cancer detection. Better control of blood pressure and lipids. Environmental changes that reduce exposure and support movement. Each adds a layer of maintenance. The result may be a later onset of several diseases rather than biological immortality. That outcome would still be profound. A 5-year delay in frailty, dementia, cancer, and cardiovascular disease across a population would change millions of lives. It would not need anyone to reach 200. The most important progress may arrive without a dramatic record holder. It may appear as an older population walking farther, recovering faster, and spending less time disabled. Health span is difficult to advertise because it has no single miraculous image. It is the preservation of ordinary capacity, the body continuing to meet the day. The body continuing to meet the day is also the body that returns to night. The maintenance described at the beginning has never stopped. DNA repair enzymes still inspect strands. Ribosomes still build proteins. Lysosomes still receive damaged material. Bone continues its slow exchange. Immune cells move through tissue. The kidneys adjust water and salts. Muscle responds to earlier use. The brain changes its pattern as waking attention becomes less demanding. These processes do not all begin with sleep. Some follow meals. Some follow exercise, injury, infection, temperature, or darkness. Some are guided by circadian clocks. Some continue at nearly constant rates. The body contains rhythms nested inside other rhythms. A heartbeat completes in less than a second. A sleep cycle unfolds across roughly an hour and a half. Red blood cells remain in circulation for months. Bone remodeling continues across longer intervals. Immune memory can survive for decades. A mutation acquired by one stem cell may remain unnoticed for years before its descendants expand enough to matter. Aging links these timescales without belonging to any one of them. No individual cell experiences the number written on a birthday. It experiences oxygen, nutrients, mechanical force, hormones, neighboring cells, damage, and chemical signals in the present moment. It responds locally. Across trillions of local responses, an organism changes globally. This is why no one has found a single place where aging resides. Lengthening a telomere does not restore every damaged protein. Removing one senescent population does not correct every mutation. Resetting methylation does not dissolve arterial calcium. Improving mitochondrial function does not rebuild every joint. Strengthening muscle does not restore every sense. Each intervention can matter without becoming complete. The organism is connected enough for one failure to spread and connected enough for one benefit to spread. Walking can influence muscle, blood vessels, glucose regulation, balance, and mood. Treating hearing loss can change communication, activity, and cognitive demand. Vaccination can preserve reserve by preventing an infection. Controlling blood pressure can protect the brain, heart, and kidney at once. This network is what makes aging difficult to reduce to one drug. It is also what makes health span modifiable. The most accurate image may not be a clock running down. A clock has one mechanism and advances through one scale. Aging is closer to a landscape crossed over time. Genes shape parts of the terrain. Childhood, illness, injury, work, housing, pollution, food, movement, relationships, and medical care alter the route. Recovery may return the body near an earlier path or establish a new one. Chance remains present at every stage. Two people can reach the same chronological age through very different landscapes. Their useful interventions will not always be identical. A therapy that helps insulin resistance may offer little to a lean person with an immune disorder. Calorie restriction may improve one metabolic state and worsen frailty in another. Suppressing mTOR may help one process while limiting muscle adaptation. A senolytic may benefit one tissue and interfere with healing in another. This is not evidence that nothing works. It is evidence that the target is an organism rather than an abstract average cell. The same caution applies to biological clocks. A clock can estimate a pattern. It cannot predict every future infection, fall, mutation, treatment, or recovery. It cannot convert a population relationship into one certain lifespan. Gene Kalman's 122 years show what one documented human body achieved under one history. Greenland sharks show that vertebrate tissues can persist for centuries. Naked mole rats show that a small mammal can age far more slowly than expected from size. None supplies a transferable switch. Long-lived species expand the map of biological possibility. Human trials determine which paths are safe for humans. That bridge from possibility to treatment is built through time, controls, replication, and failure. Many interventions that alter cells fail in animals. Many that extend mouse life fail to improve human health. A dose becomes toxic. A biomarker proves non-causal. A pathway behaves differently across tissues. A laboratory model omits the disease or environment that matters. Failure can still refine the map. The history of aging science has repeatedly become more accurate by losing a simple explanation. Cultured cells were assumed to divide indefinitely until Hayflick showed a limit. Telomeres helped explain the limit, and cancer showed why escaping it can be dangerous. Free radicals appeared to offer one broad mechanism until signaling biology and disappointing antioxidant trials revealed a more complex redox system. Senescent cells appeared to be inert debris until development and wound healing showed useful functions. Young blood appeared to contain rejuvenating factors until circulation experiments showed the importance of dilution, shared organs, and harmful old signals. Epigenetic clocks appeared to provide one biological age until different clocks disagreed, and clinical validation exposed the difference between prediction and mechanism. The loss of simple answers does not leave nothing. It leaves a stronger conclusion. Aging is biologically modifiable. It is not yet controllable as a whole. Some causes of premature death can be removed. Many age-related diseases can be prevented, delayed, or treated. Strength and aerobic capacity can improve late in life. Cataracts can be replaced with clear lenses. Joints can be rebuilt. Vaccines can compensate for older immunity. Experimental pathways can extend lifespan in animals. At the same time, no drug, supplement, plasma infusion, stem cell procedure, or reprogramming treatment has been shown to make healthy human bodies broadly young again or to extend the verified maximum human lifespan. Optimism belongs beside restraint. The outcome that matters most is not whether one molecular number moves downward. It is whether a person can continue to move, think, communicate, recover, maintain relationships, and live with independence and tolerable pain. Those abilities are distributed across the body and across life. They resist reduction to one assay because life itself is integrated. The body tonight already contains several kinds of time. Some neurons may have been present before birth. Proteins in the center of the eye lens were formed during development. Immune cells carry memories of infections and vaccines. Bone contains regions laid down at different ages. Scars preserve repair after injury. Mitochondrial DNA retains the remnant of an ancient bacterial partnership. Aging does not erase the body's earlier states completely. An older muscle still uses pathways that built the younger one. An older immune cell can still recognize a past pathogen. An older brain still alters connections. Plasticity narrows and remains. The word decline can hide this continuity. Older organisms are not younger organisms with only missing pieces. They contain decades of adaptation, exposure, learning, repair, and survival. Biological losses are real without making every direction negative. Knowledge and practice judgment can deepen while processing speed changes. Social roles can expand after reproduction ends. Care, memory, teaching, and continuity are not represented by a methylation clock. The science of longevity should preserve years because the years themselves matter. The question of why bodies grow old can now be answered without choosing only one mechanism. Bodies grow old because molecular damage is unavoidable and repair is not perfect. Because cells must limit division to reduce cancer risk. Because pathways for growth, immunity, reproduction, and repair create benefits at one stage and costs at another. Because the force of natural selection weakens against many late effects. Because proteins, mitochondria, stem cells, extracellular structures, organs, and communication networks change together. Because environment and society leave cumulative biological marks. Because chance acts within every layer. Aging emerges where these processes intersect. The quietest fact is how long the body resists that intersection. Damage did not wait until old age. DNA has been altered and repaired throughout life. Proteins have misfolded and been removed. Cells have become dangerous and been stopped. Pathogens have entered and been contained. Bone has cracked microscopically and been rebuilt. Blood cells have disappeared and been replaced. A body reaches old age not because damage suddenly began. It reaches old age because maintenance succeeded repeatedly before becoming less complete. Aging is evidence of imperfect repair. Longevity is evidence of how much repair accomplished. That is the return to the still body at the beginning. One night will not reset every epigenetic mark, lengthen every telomere, clear every senescent cell, or restore every stem cell niche. Sleep is not a reversal treatment. It is one ordinary state inside the regulation that has supported the organism for years. Heart rate changes. Hormonal signals shift with the sleeping state. Immune signaling shifts. Memory is reorganized. Muscle continues responding to earlier use. Cellular work proceeds without entering awareness. Conscious attention cannot improve the accuracy of a repair enzyme. It cannot select the next mitochondrion for mitophagy or calculate the correct inflammatory signal. There is no need to supervise the process. Research laboratories can continue following cells, mice, biomarkers, and clinical trials. Demographers can continue validating the oldest lives. Medicine can continue learning how to preserve function. The body in this room has only the immediate work of continuing through the night. The heart does not need to know the Gompertz curve. A chromosome does not need the word telomere. A mitochondrion does not know that its ancestor was a bacterium. No birthday changes within the next breath. Aging is slow enough to disappear from one ordinary moment. The maintenance systems remain active beneath stillness. They will not make the body permanent. They have already made it enduring. The years do not need to be counted tonight. Sleep well. Good night.